Welcome to your guide to the biology domain of the ATI TEAS exam, one of the largest domains, 18% of the content. In this video, we'll cover the cell and how it works, how DNA codes for proteins, how traits pass parent to child, the 4 macromolecules, the microbes that cause disease, and remember, our website gives you free exam guides and practice questions to help you ace your ATI TEAS exam. You'll find the link right in the description below, so go ahead and check it out. Study smarter, not harder, and you'll be ready for test day. All right, let's get started. Let us start with the cell, the basic unit of life. Every living thing is made of cells, from single bacteria to the trillions that build a human body. The TEAS wants 2 broad types. Prokaryotic cells, like bacteria, are small and simple with no nucleus. Eukaryotic cells, like ours, are larger and packed with compartments. Inside a eukaryotic cell, picture each organelle as an employee with one job. The nucleus is the control center, holding your DNA and issuing instructions. The nucleolus inside it builds ribosomes. Ribosomes are the protein factories reading genetic messages and assembling amino acids. Some float free in the cytoplasm, others attach to the rough endoplasmic reticulum. That brings us to the cell's manufacturing district. The rough endoplasmic reticulum, studded with ribosomes, makes proteins for export. The smooth endoplasmic reticulum builds lipids and helps detoxify drugs. From there, proteins travel to the Golgi apparatus, which I think of as the post office. It modifies, packages, and ships each protein where it belongs. Here is the organelle I would memorize first because it appears constantly. The mitochondria, the powerhouse of the cell, is where most of your ATP, the energy currency, gets made. Its inner membrane folds into ridges called cristae, adding surface area for energy reactions. A favorite exam fact is that mitochondria carry their own DNA. A few more workers round out the team. Lysosomes are the recycling centers, full of enzymes that break down worn parts and invaders. Peroxisomes handle fatty acids and neutralize toxins. The cytoskeleton is a protein scaffold, giving the cell shape and acting as internal railways. And the plasma membrane wraps it all, deciding what enters and leaves. What about plant cells? They have everything an animal cell has, plus 3 extras. A rigid cellulose cell wall for support, chloroplasts, where photosynthesis captures sunlight, and a large central vacuole storing water. A classic trap asks which feature is unique to plants on the exam. The answer you want is the cell wall. Let me ask you this. How does a cell move things across that membrane? 2 ways. Passive transport needs no energy and includes diffusion, where molecules drift from crowded to less crowded areas, and osmosis, the diffusion of water. Active transport spends energy to push molecules against their gradient. The sodium-potassium pump is the classic example. This sets up tonicity, a favorite exam topic. Picture a red blood cell dropped into pure water, a hypertonic solution. Water rushes in and the cell swells until it bursts. Drop it into very salty water, a hypertonic solution, and water flees outward, so the cell shrivels. In an isotonic solution, concentrations match and the cell stays normal. Cells divide following the cell cycle. The cell grows, copies its DNA during the synthesis phase, prepares, then splits during mitosis. Mitosis runs through 4 stages in order: prophase, metaphase, anaphase, telophase. A memory trick I like is the phrase "pass my anatomy test," giving those 4 in sequence. Mitosis yields 2 identical daughter cells. Meiosis is the other division, and students mix these up constantly. Meiosis makes sex cells, sperm and eggs, through 2 rounds of division that produce 4 cells with half the chromosomes. It also shuffles genes, creating variety. And remember how cells get energy. Cellular respiration breaks glucose down through glycolysis, the Krebs cycle, and the electron transport chain. Plants run the whole thing in reverse through photosynthesis. Inside chloroplasts, they capture sunlight to turn carbon dioxide and water into glucose and oxygen. So respiration burns sugar for energy, while photosynthesis builds sugar using light. They are mirror images, and the oxygen plants release is the very oxygen your mitochondria need. A neat loop the exam tests. What we just covered is exactly the kind of material waiting in our free practice question bank. These are not throwaway questions either. They are written to test the deep understanding the real TIS demands, mixing organelles, transport, and cell division the way the actual exam loves to combine ideas. Go try a few. Now this is where it gets interesting. Why does your body bother with DNA at all? Because DNA is the master instruction manual, and proteins are the workers that run your cells. The whole flow fits one phrase examiners adore. DNA makes RNA, and RNA makes protein. Lock in that direction and this section gets easier. Let us look at DNA, a twisted ladder built from units called nucleotides. Each has a sugar, a phosphate, and one of 4 bases: adenine, thymine, guanine, cytosine. The pairing rule is everything. Adenine bonds with thymine, guanine with cytosine. A handy mnemonic links apples in the tree with cars in the garage. RNA is DNA's working partner, and 3 differences set them apart. RNA is single-stranded, not a double helix. Its sugar is ribose instead of deoxyribose, and it swaps thymine for uracil. There are 3 kinds worth knowing. Messenger RNA carries the message, transfer RNA hauls amino acids, and ribosomal RNA builds the ribosome. The first half of the flow is transcription, copying a DNA gene into messenger RNA inside the nucleus. RNA polymerase unzips the gene, reads one strand as a template, and builds a matching RNA copy. Think of it like photocopying a single recipe from a cookbook you never take off the shelf. That copy then leaves the nucleus. The second half is translation, where the ribosome reads that message and builds the protein. The message is read 3 bases at a time, and each triplet is a codon. Every codon names one amino acid. Transfer RNA molecules arrive carrying the right amino acid, matching their anti-codon to each codon. The chain grows until a stop codon. A couple of codon facts earn easy points. There are 64 possible codons, more than enough for the 20 amino acids, so several codons can spell the same one. The codon spelled AUG is special. It is the start codon beginning every protein, and it codes for the amino acid methionine. 3 codons signal stop. Before a cell divides, it must copy all its DNA, a process called replication. Here is the detail examiner's test. Replication is semi-conservative. Each new double helix keeps one original strand and one brand new strand. Helicase unzips the 2 strands, then DNA polymerase reads each side and adds matching bases. The result is 2 identical molecules. Sometimes the copying slips, and we call that a mutation, a change in the DNA sequence. Swap one base and you have a substitution. Add or remove a base and you trigger a frame shift which scrambles every codon downstream, usually with serious effects. Mutations can be harmful, harmless, or occasionally helpful, the raw material of evolution. One more piece completes the picture, which is how proteins fold. The primary structure is the amino acid sequence, like beads on a string. That string coils into secondary structures, folds into a tertiary shape, and sometimes several chains join as quaternary structure, the way hemoglobin uses for The rule that matters most is that shape determines function. So why does shape matter so much on the exam? Because if a protein loses its shape through denaturation from heat or wrong pH, it stops working entirely. That single idea connects to enzymes, fever, and even cooking an egg. When a question describes a protein failing after heating, denaturation is almost always the answer. I know we are covering a lot of detail, and you might wonder how to practice applying it. That is exactly why I built those free practice questions I mentioned. They let you work through problems that combine transcription, translation, and mutations together, just the way the TEAS likes to test real understanding. Go put them to work. Picture two brown-eyed parents who have a blue-eyed baby. Surprised? By the end of this section, you will predict outcomes like that with confidence. We owe these rules to Gregor Mendel, who cracked inheritance using pea plants in the 1860s, long before anyone had seen DNA. The TEAS asks you to predict inheritance. First, the vocabulary, because the questions live or die on it. A gene is a unit of heredity, and an allele is one version of that gene. You carry 2 alleles per gene, one from each parent. If they match like big B and big B, you are homozygous. If they differ like big B and little b, heterozygous. 2 more pairs finish the toolkit. Genotype is the genetic code you carry, the letters themselves. Phenotype is what you actually see, like eye color. And alleles come in 2 flavors. A dominant allele, written capital, shows itself whenever present. A recessive allele, written lowercase, hides unless doubled up. So either brown combination still gives brown eyes. Now, back to those brown-eyed parents. The tool that solves it is the Punnett square, a simple grid. Put one parent's alleles across the top, the others down the side, and fill each box. Cross 2 heterozygous parents, and you get 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive offspring. So what do those 4 boxes mean? 3 of the 4 carry a dominant allele and look brown, while 1 has 2 recessives and looks blue. That is the classic 3-to-1 ratio. It explains our surprise baby. 2 brown-eyed parents, each a hidden carrier, have a 1 in 4 chance of a blue-eyed child. A few cross types recur on the test. The monohybrid cross tracks 1 trait and gives that 3-to-1 ratio. The test cross pairs a mystery individual with a homozygous recessive partner to reveal a hidden genotype. The dihybrid cross follows 2 traits and gives the 9-to-3 to 3-to-1 pattern. Recognize those numbers. But here is the part most students miss. Not every trait obeys simple dominance. In incomplete dominance, the heterozygote blends, so a red flower crossed with a white one yields pink. In codominance, both alleles show fully. Blood type is the perfect example. The A and B alleles are codominant, so one of each produces type AB blood. 3 more patterns remain. Multiple alleles means a gene has more than 2 versions in a population, like the 3 blood type alleles, though you inherit 2. Polygenic traits like height and skin color blend many genes into a smooth range. And sex-linked traits sit on the X chromosome, so color blindness strikes more men. Let me walk you through a question they genuinely ask. A father has type AB blood, a mother type O. What can their children be? Dad passes either A or B. Mom can only pass O, which is recessive. So every child gets 1 letter from dad and an O from mom, landing as type A or type B. Underneath all of this sit Mendel's 3 laws. Segregation says your 2 alleles separate, so each gamete carries just 1. Independent assortment says different genes sort into gametes independently. Dominant says the dominant allele masks the recessive when both appear. If you remember only one, make it segregation, since splitting alleles is what every Punnett square is built on. If Punnett squares and blood type crosses feel tricky right now, do not worry. That is completely normal, and it is exactly why I created that practice question bank. Working through cross after cross is how this clicks. The questions are free and they build the pattern recognition that turns genetics into one of your easiest sections. Next come the macromolecules, the 4 big molecule families that build every living thing. The cleanest way to hold them is carbohydrates, lipids, proteins, and nucleic acids. For each one, the exam wants the same 3 things: the building block, the main jobs, and a few examples. Nail that pattern and all 4 become predictable. Carbohydrates come first, and their building block is the monosaccharide, a simple sugar like glucose. Join 2 and you get a disaccharide such as table sugar. Join many and you get a polysaccharide like the starch plants store or the glycogen we store in muscle and liver. Their main job is quick energy, though cellulose gives plants structure. Lipids are next, and the one thing they share is hating water. Their jobs are impressive: long-term energy storage, insulation, and forming every cell membrane. 3 types matter. Triglycerides are your fats, saturated ones solid like butter, unsaturated liquid like oil. Phospholipids build membranes. Steroids include cholesterol and hormones. Notice fat stores twice the energy of carbohydrates. Proteins we have met, but they earn a spotlight here. Their building block is the amino acid, and 20 combine in endless orders linked by peptide bonds. The jobs are the widest of any molecule: enzymes, antibodies, hemoglobin carrying oxygen, and structural collagen and keratin. When a question lists a worker molecule, suspect a protein. The 4th family is nucleic acids, and you already know the stars, DNA, and RNA. The building block is the nucleotide, that sugar, phosphate, and base trio. Their job is storing and carrying genetic information. Here is a detail worth a point. ATP, the very energy molecule we met inside the cell, is itself a nucleotide. Everything connects. Why should a nurse care about all 4 families? Because they are what food, bodies, and many medicines are made of. The carbohydrates on a nutrition label, the fats flagged for heart health, the protein in a muscle, and the DNA targeted by some drugs are these exact molecules. This is the chemistry of patient care. Now zoom in on enzymes, which get their own questions. An enzyme is almost always a protein that speeds a reaction by lowering the energy needed to start it. The molecule it acts on is the substrate, binding at a pocket called the active site. Picture a lock and key, or a glove fitting a hand. 2 enzyme facts win points. First, they are picky about conditions, working best at a specific temperature and pH. Push them too hot or too acidic and they denature and quit. That same shape lesson. Second, enzymes are reusable. They are not consumed in the reaction, so one enzyme works over and over. That reusability is a common trap. Here is how I would lock all 4 families in for test day. Match each building block to its family like flashcards. Monosaccharide means carbohydrate, amino acid means protein, nucleotide means nucleic acid, and lipids stand apart with no single block. The TEAS loves asking which building block goes with which molecule, so that drill earns quick points. Remember that mastering these concepts takes practice, not just listening. The free practice questions are built specifically to take what you're learning here and turn it into exam-ready skill. A few minutes of questions after this video will do more for your score than rewatching ever could. Keep that link close. Let me start our last concept with a mistake that costs real points and real patience. Antibiotics do nothing against viruses. If you remember only one thing from this section, make it that. Microorganisms are the tiny life forms, and several types cause disease. As a future nurse, telling them apart shapes how every infection gets treated. Bacteria lead the list. They are single-celled prokaryotes, meaning no nucleus, and they have a cell wall. You can sort them by shape, round coxy, rod-shaped bacilli or spiral spirilla, and by their Gram-Stain reaction. Crucially, bacteria are alive and reproduce on their own, which is why antibiotics can target them. Strep throat is bacterial. Viruses are the real troublemakers. A virus is not even truly alive. It is just a scrap of DNA or RNA wrapped in a protein coat with no way to reproduce by itself. It must hijack one of your cells and force that cell to build copies. That dependence is why antibiotics fail and why we use antivirals instead. 3 more groups deserve a mention. Fungi are eukaryotes, including single-celled yeast and multicellular molds, causing things like athlete's foot and yeast infections. Protozoa are single-celled eukaryotes, and the big one is the malaria parasite. Then there are helminths, the parasitic worms, and prions, misfolded proteins that trigger rare brain diseases. So how do these microbes actually make us sick? A few ways. Some invade and kill cells directly, the way HIV destroys immune cells. Many bacteria release toxins, poisons that damage tissue, like those behind tetanus and cholera. Sometimes our own immune overreaction does the harm, as in life-threatening sepsis. And disturbing helpful microbes lets opportunists take over. How microbes spread is a favorite question, so learn the roots. Direct contact means touching an infected person. Droplet spread travels a few feet through coughs like the flu. Airborne particles drift much farther like tuberculosis and measles. Vehicle transmission rides in contaminated food or water. Vector transmission hitches on an animal like malaria from mosquitoes. Our defenses come in layers. The immune system fights from inside while medicines help from outside. Antibiotics target features unique to bacteria, like their cell walls, which is why penicillin works on them but never on a virus. Antivirals disrupt viral life cycles. Antifungals hit fungal cells, and vaccines train your immune system to recognize an invader fast. That brings us to antibiotic resistance, a big deal in medicine. When you take an antibiotic, it kills the weak bacteria but spares any with a mutation that resists it. Those survivors multiply and the drug stops working. The main drivers are overusing antibiotics, especially for viral colds, and quitting a prescription early. Finish your course. One more idea matters for nursing. Not all microbes are enemies. Trillions of helpful bacteria, your normal flora, live in your gut and on your skin, aiding digestion and crowding out invaders. Antibiotics can wipe them out along with the harmful ones, which is how a stubborn gut infection takes hold after treatment. Balance matters. Let me give you the comparison the exam returns to again and again. Bacteria are bigger, alive, often have cell walls, reproduce alone, and answer to antibiotics. Viruses are tiny, not truly alive, need a host to copy themselves, and shrug antibiotics off. Sort any pathogen into that bacteria versus virus split, and you earn reliable points. As we wrap up this overview, remember that understanding these ideas is only the first step. Real mastery comes from applying them, and that is where those free practice questions become invaluable. They cover cells, genetics, macromolecules, and microbes, all completely free and designed specifically for TEAS success. The link is waiting in the description. Look how far you have come in one sitting. You can walk through a cell and name what each organelle does. You understand how DNA becomes RNA and then protein. You can set up a Punnett square and predict a child's odds. None of that was memorizing for its own sake. It was understanding. Notice how tightly these topics weave together. Proteins built in translation become the enzymes and antibodies of the macromolecule chapter. The mutations that change DNA are the same ones driving antibiotic resistance in bacteria. Cell membranes are made of the phospholipids we covered as lipids. The TEs loves questions crossing these lines, so seeing connections lifts your score. Here is my honest promise to you. If you review this material a few times and pair it with real practice, this entire biology section becomes one of your strongest. So do not forget to grab those free practice questions from the link in the description. With today's understanding plus that practice, you will walk in prepared. You have got this. Biology is not about being a genius. It is about understanding a handful of core ideas well enough that the answers feel obvious. You just spent real time building exactly that understanding. Now go reinforce it with practice. Trust your preparation and walk into that exam room ready to show what you know. Good luck. You've reached the end of our biology review. If you'd like free exam prep, head over to our website using the link in the description, where hundreds more practice questions and study guides are waiting for you. We'd genuinely love to help you keep learning. Stay confident and feel completely ready when exam day finally arrives. Thanks for studying with us today. See you next time.